School 2021. So I can see that most of you are back now from a short break. So yeah, make sure that you open your video camera and then mute your microphone as usual. The sessions are all recorded. So we are already starting our recording now. Um, Yes. So for the next session, the next module 6, which is on integrative modeling of biomolecular complexes toward interactome modeling, we have distinguished lecturer here, Professor Alexander Bonvin. Hello, Professor Bonvin. How are you? You can unmute, yes.
Yes.
Good. So I think it's
All
already.
good. Pleasure to be here.
Yes, great. We're already very excited to have you here. So it's still morning time there.
Yes, just past 9. Just had my coffee, ready to go.
Oh, I can see you're very excited already. So before we start, I would like to introduce you shortly for the students here. Professor Bonvin studied chemistry at Lausanne University, Switzerland, and obtained his PhD at Utrecht University in the Netherlands. And he was appointed as full professor of computational structural biology in 2009 there. He is currently scientific director of the Baywood Center for biomolecular research, and his group developed and operates the Haddock Integrative Modeling Platform. So full bio of Professor Bonvin is in the program module, so you can refer to that for the students. Without further ado, the floor is yours, Professor Bonvin. Have a good session. Thank you.
Thank you very much. So let me share my screen. Okay, welcome for this afternoon session for you, morning session for me, about integrative modeling of biomolecular complexes. So I'm Alexandre Bonvin. I'm based in Utrecht in the Netherlands, working within the Baywood Center, where we have a focus on biomolecular interactions. And it's really my pleasure to be here today to speak to you about this topic. I have with me, you might have seen his face in Zoom, Rodrigo, who is a senior researcher in my lab, and Rodrigo is going to monitor the chat. So if you have questions, you can ask questions during the presentation already. And I will take probably questions during the presentation after I'm changing topics. And then later on during the tutorial as well. So what are we going to talk about for the coming hour or a little bit less of an hour to leave enough time for the tutorial? So I'm going to give you a general introduction on the topic, and then I'm going to discuss the problem of docking, macromolecular docking, and generating models of complexes in more general aspects so that you get a feeling of what are the steps and what is involved and what are the challenges. And then I will come more to the specific of Haddock, which will be the focus also of the tutorial. And at the end, I will show you a very short application example of what you can do and show some highlights of more recent developments. So what's the topic? You're all aware, so we have the genomic information, which encodes for the proteins, which brings us to the level of the proteome. And proteins are doing their work by interacting with other biomolecules. And it means that the functional level of a cell are complexes, are interactions. And you see here a zoom in such a complex where you see details of the interface, hydrogen bonding, and understanding these interactions at atomistic level is very important because often when mutations occurs, not only within the protein, which might change the protein fold and stability, but also at the interfaces, this is where things go wrong. So many mutations in interfaces are basically affecting the communication network of cells. So by understanding those complexes at the free, at atomistic level, you can get insight into, say, disease causing mutations. You might try to engineer molecule for better binding of better materials. And of course, starting point for drug design. If you want to design small molecules that are going to prevent or restore interactions, you need to understand how those take place. So this brings me to the structural biology of interactions. So how can we study complexes, biomolecular complexes? And on the right side, you see a list of experimental methods. So this is the world of experiments, where the three main methods to get atomistic structures of biomolecules are X-ray crystallography, the oldest one, nuclear magnetic resonance, and since several years now, cryo-electron microscopy, which is really a star method now in structural biology. So those three methods allow you to get really full atomistic pictures of biomolecules. But next to those methods, there is a plethora of other experimental methods that are going to give you pieces of the puzzle, but they will not allow you to solve completely the structure from scratch. So example are mass spectrometry, which might provide you information about, interface between molecules, possibly, excuse me, some distances. Threat experiments are also giving you some not very accurate distances, but it's still valuable information. And there are other experiments like small angle scattering, X-ray or neutron scattering, which is going to provide you information about shape of complexes. So if you have this kind of lower resolution information, what you will need to do is to go into the world of computations to basically complement the scarce experimental information with computer models and with computations to generate models of those 3D structures. Now, if we look into the computational side of things here, there are different ways of looking at complexes. So you might try to generate a complex simply by homology modeling, provided there is the structure of another complex available in the protein database. Threading will be a related method, basically. And molecular dynamics, you've heard about that. You might try to fold proteins. But for today, the topic of my talk and also the tutorial will be about docking. So docking in a nutshell, so given the structure of two molecules in this particular example, but you have to realize that many complexes, biomolecular complexes, consist of more than two molecules. So we'll have also to be able to build assemblies consisting of several molecules, which increase the complexity of the problem. But say, given the structure of two molecules, can we predict how they interact? So what is the problem here? So if we were to assume that the proteins are rigid, which is not the case, what we have to sample is a 6-dimensional space, because you can fix one molecule at the origin of your space. And what you have to do for the second molecule is to sample all possible translations. So in 3D, these are 3 translations, and also sample all possible rotations. For each rotation that you sample, you have to sample all translation. So for 2-molecule docking, the problem is the 6-dimensional search problem if the molecule are rigid. So remember, you can fix one molecule at the origin of space. You don't need to translate, rotate, and you rotate and translate the other one. And then you can try to figure out what if I had one molecule, what will be the dimensionality of the problem. Now, how do you measure if the solutions are good or bad? We use classical, the most software use a variety of things. You see here 2 energy terms. So this is electrostatic, Coulomb potential, and you see here Van der Waals, Lennard-Jones potential. So with those, you can measure the complementarity, energetic complementarity of the interfaces, but you can also use geometric criteria. You can use conservation criteria. You can use statistics. We're going to come back to some of those. So what do we have to do? So we have first so you have here a very simple plot where you see on the x-axis some kind of conformational or interaction landscape. So this will be the interaction landscape between the molecule. And on the y-axis, you have some kind of energy function that measure how good the model that you generated basically fits this energy function. So we have this landscape that we have to sample. So we need methods to generate a lot of different models of the interactions. And then we need to score those models. So in docking, we distinguish sampling and scoring. And scoring means using some kind of score function, energy, statistical potential to distinguish what are the good models from the bad one. And the assumption here is that, say, the models that will be at the with the best score, the lowest energy will correspond to the native solution of this complex. Now, my talk is about integrative modeling. So you might have data at hand, and those data might be experimental or bioinformatics data. So you can decide to use those data in two ways. So you could use the data to bias the sampling, so bias the search of this interaction space, or you can use the data in the scoring. So these are different strategies, and actually you can use the data in both. So we distinguish, data-driven sampling from only data-based scoring. So if you don't use data during the sampling, what you are going to do is a global search. You have to search the entire interaction space, and you're going to generate a very large number of models, and then you will have to score them. If you have data, and if you can use the data during the sampling process, what you're going to do is to concentrate the search on a specific region of space, which has advantage because you can spend more time searching, hopefully in the right region of space. But this also has dangers, because if the data are bad, if you have wrong data, you might search completely in the wrong region of the interaction space and never find the right solution. Okay, so there are positive aspects about using data to bias the search, but there are also dangers in using the data. So you have to have trust in the data, basically, if you're going to follow this route. Now, these days, when we speak of integrative modeling, we really speak about the combination of modeling technique, computational techniques, together with different information sources to guide the modeling. So what can you use when it comes to interactions? There's a lot of different methods. NMR, nuclear magnetic resonance, allows you to, for example, identify which region on a protein surface is affected by the binding. So you see here a very simple spectrum. One residue called A is not affected when you add different concentration of a, say, small molecule, but you see that B is affected because its signal is shifting. So this will be interpreted as B must be involved in the binding process while A is not involved. So this is going to give you information about binding sites, but it's not going to tell you how things are binding. Another source of information will be, for example, hydrogen-deuterium exchange, which is usually detected by mass spectrometry. These are changes of masses, where you dissolve your proteins into D2O, and then all the protons that can be exchanged will be exchanged by deuterons. And if you do that for the single proteins and the complex, and you look at the differences, the regions that are protected from exchange might point to interfaces in your complexes. Cross-linking MS, a very popular method these days, very powerful. So here you use small molecule chemicals that are going to attach to a specific group on the surface of your proteins. Typical groups that are targeted are lysine side chains. And then you have to detect those by MS, so you break your protein into peptide, do the detection, and you will find from time to time two peptides originating from different proteins connected by this crosslink. So this is providing you distance information between specific residues. PREs, EPR, PREs, EPRs are magnetic-related experiments. FRET will also provide you some kind of distance information. NMR can provide you information about orientations. Bioinformatic, very powerful these days. By looking at conservation of amino acid, you might detect binding sites, but also coevolutions. Probably you're all aware of the success of Google with AlphaFold 2 in the recent CASP experiment. And here you are not only predicting interfaces, but you might be predicting contact as well. And contact can be introduced as some kind of distance restraints in the modeling process. Doing mutagenesis experiments in a biochemical lab basically will also point you to residues that are crucial for the binding. This is information. And then the kind of low resolution shape information that you can get from small angle scattering, but also cryo-EM. You have to realize that cryo-EM, although it has achieved fantastic progress over the years, you will not always reach a resolution which is necessary to build a model from scratch. So there will always be cases where the resolution is limited. You cannot build the structure of the complex from scratch, but you can use modeling to build those models. So a combination of those data together with computational modeling is what we call integrative modeling. Here are some reviews that we wrote on the topic. I just want to point you also that in proteins, they are every 2 years, a special issue dedicated to Capri, and Capri is a critical assessment of predicate interaction. It's kind of a blind experiment to measure how well the field is doing in predicting protein complexes, while Cas is the equivalent of Capri, but for the prediction of structures. So this was my general introduction to the topic. So now let's dive more into specifics of docking. So what are the choices that you have to make when you're going to start using docking approach computational methods to build complexes? So first you have to think, how do I represent the system? Okay, so this is going to define how you do the search. Then you have to worry about the sampling. So I already explained you that if for two molecules, if they are rigid, what we have to sample is 3 rotations and 3 translations systematically. So rotations are on 3 axes and translation in 3D. But if you want to describe conformational changes, flexibility, you will have to worry about that. And of course, biomolecules are flexible. Flexibility dynamic is an interesting part of their function. You will have to worry about how to distinguish models, good from bad. And if you have data, how are you going to include those data in the modeling process? So let's move now into the representation of the systems. The first docking software, and this is, we are speaking now about, let's say the 70s, early 70s, the work of Joël Janin and Shoshana Bodak, was using an explicit representation of the system where we'll be handling the coordinates of all atoms in 3 dimensions. So the search was in real space, in Cartesian space, and this also made at the time the software quite slow. But basically you have to sample those rotations, the translations in 3D, and then hopefully get a model of the complex. Now, what has advanced a lot the field was to switch from this explicit representation to what is called grid-based representation. So what you do here is that you put a grid onto your system, onto your molecules, with a specific spacing between the grid points. You see here an example of a 1 angstrom spacing, and you see a tyrosine sidechain here. And what you are doing is now you are transforming the atomistic description of all the coordinates of your system into a grid representation, where you're basically assigning properties to the grid points, or to the voxels actually in grids, which are the volume, the cubes. So you will describe the surface of the protein. These are these bluish grid points, and then the inside will be gray points. And then you forget about the atoms, and you only work with grids to do the search. And what you want to do when you do the search is to maximize the overlap between the blue regions, because it means complementarity of surfaces. And you want to minimize the overlap between gray regions in this example, because this will mean clashes between atoms. And to do this search, you can do it in real space. Some software like Bigger used to do that. But most docking software are doing the search in Fourier space. Because once you start working with grid, equally spaced points, Fourier transformation, fast Fourier transformation techniques become very efficient, which means that you can do the search, translational search in this case, using the FFT methods. Now, you also find representations that are a mixture of grids and explicit representation. This is used more in the small molecule fields, where, for example, in Autodoc, which most of you must know, the protein is basically represented onto a grid, or actually the energetic contribution of the protein is represented in the grid, and the ligand is explicitly described. So we describe the position of the atoms of the ligand, but this ligand moves into an energetic grid of the protein. So it's a mixed representation. But we have even more ways of describing proteins, and the next methods, which is also a very interesting, a very fast way of doing docking, is to replace the atoms by a representation of the surface. When we use grid docking, we also represent explicitly the surface, basically. But here, what you do, you take a linear combination of spherical harmonics, and spherical harmonics are mathematical function, trigonometric function. You see them represented here. So the chemists among you might recognize these are orbitals, electronic orbitals, but these are simply mathematical function. And actually, we use the same spherical harmonics to represent the electronic orbitals. And by taking a linear combination of those functions, so we take 15 different functions, and they are of different order from -1 to 1. So it's 45 terms in this function. And optimizing the parameters here, you can go from atomistic representations to a basically surface representation of your proteins. If you were to look at all the atoms in this system, you might have 10,000 coordinates that you have to deal with, and now you replace 10,000 coordinates by only 45 parameters, basically. So it's a huge reduction of the complexity of the system, and it means that you can perform the search very quickly. And now the search, if you work with spherical harmonics, you can also use fast Fourier transformation techniques, but in this case, you are going to use it to do the rotational search, and you have to explicitly do the translational search. And the last method I just want to mention here for completeness is yet another way of looking at the problem, and this is thinking in terms of puzzles. Okay, so we are going to now decompose the surface of your protein into puzzle pieces. So you do a triangulation of the surface and you identify different pieces of the puzzle. And then when you solve, if you solve a puzzle, you're not taking all the pieces at the same time and trying to solve the puzzle in one go. That's a very complex problem. But what you do is you select one piece of the puzzle and then you look for a match that will fit. And in doing docking in this puzzle-based approach, we do exactly the same. So we decompose the protein in puzzle pieces. So we have a list of puzzle pieces, and then we search for matches in the other protein. And once you have a match, you reconstruct only the full complex. And this is implemented in PatchDock, which is another docking software, which is extremely fast. Any questions at this time on the...
So far, so good.
So far, so good. So these were the different ways of representing the system. So now let's see how we can search the space. I already mentioned several times we have to search this 6-dimensional space for 2 molecule translation and rotations. So this is so this might take quite some time depending on the size of your proteins. And for each model that you generate, so each sampling of rotation and translation, you also need to calculate a score to identify or predict if this is a good model or not. And depending on the scoring function that you use, this might become very time-consuming. Now, I already explained if you use grid representations, software like ZDoc, ClusPro, Gram, FTDoc use grids, you can do the search, the translational search in using fast Fourier transformation techniques. If you use spherical harmonic representation like Hex is doing, you can do the rotational search in the using FFT methods. So how does it work? So you have 2 molecules. So what you're going to do is to discretize those molecules in a grid. For the largest of the 2 molecules, you only need to do this process once. So you transform from atomistic representation of the protein into a grid representation, and you associate properties to the grid voxels. So the red here will be the core, and the bluish will be the surface. For the ligands, you need to sample all the rotations, and for each rotation that you have sampled, you generate a grid. The grid will be the same size as the largest protein. So basically, the size of the problem is defined by the largest of the 2 molecules. Then you take the fast Fourier transformation of those grids, which gives you, and then you the complex conjugate, and then you correlate those in Fourier space. And what this correlation, basically what it's doing is by doing this correlation in Fourier space, you are sampling all translation in one go, pretty much. And this is what you are getting. So you're getting a correlation function as function of X and Y translation. This is a 2-dimensional example, but you will do that in 3D in real life. And for each combination of, say, X and Y translation, you get a correlation coefficient. And what you're interested in are the ones that have the highest correlation. And now if you take the inverse Fourier transformation of a specific combination of X and Y, what you are getting is the 3D structure of the complex. Okay, so that's how protein docking using FFT works. Now, in this systematic search, we're going to search all translation or rotations, which can be time-consuming. So you might do the search at different resolutions. You can use grid of different resolutions, for example, or scoring functions that are simple at the beginning. And once you start zooming in the regions of interest, you can go to more sophisticated scoring functions. At the end, it's all a question of computational time. And those models will need to be refined, but because when you do this FFT-based docking, often you end up with clashes at the interface. So the energetic of the interface will be bad, so you need to refine those models. Now, there are also the other class of methods that are energy-driven. So here we're not going to do this grid-based representation, but we have some kind of energy function that describes the interaction between the molecule, and we want to minimize this energy function. So this is an optimization problem. So here you can use methods like energy minimization, molecular dynamics, Brownian dynamics, Monte Carlo, genetic algorithms, artificial intelligence-based algorithms. So whichever optimization problem or techniques you prefer. And then you need a starting point, and then you optimize the energy of that. And you will have to repeat this process many, many times to make things work. So here you have to sample starting conformation, and this can be done randomly, for example, where you separate the molecule, apply random rotations, and repeat that many, many times. Or you could do a more systematic approach, and this is shown here for a software called ICM, where you basically put pins on the surface of your proteins. The Attract software, which is another docking software, does the same. And then you take the combination of all the pins as a starting point for an optimization, using minimization, molecular dynamics, what you want. Okay, so, but you have to repeat this optimization search many, many times. Is there a question? I see.
We have 3 questions here about sampling.
Yes.
So the first one is how we can minimize the bias when using information-driven search, or the bias or the error.
Yes. So I think I will come to that when I speak about haddock, because so far I've been speaking mainly about, say, a systematic approach that do not use data. But when I'm going to explain you how we do that in haddock, I will come to that point.
Yeah. Another one here is that you mentioned that there are 3 rotations and 3 translations for proteins. And the question is if this only applies to proteins or also to other molecule types.
To any molecule in principle. Yeah. Except if you have a benzene ring, for example, if you do a small molecule docking of a benzene, then it's a 2-dimensional problem in that case, okay, because the molecule is flat in that case. But in most cases, it will be a 3 rotations, 3 translations, yes.
Mm-hmm. And the last one here for sampling is about the grid docking and the surface patch, if these methods impact the final results of the models, the obtention of the models.
Yeah, so both those methods, but also the spherical harmonic-based methods, when you reconstitute the full atomistic representation of your molecule, you will have to refine those because you will have clashes at interface, because those methods do not allow to take really flexibility into account. And that's the next topic. So I'm going to go now into the problem of flexibility, actually, because biomolecules are flexible, they are not rigid. So we'll have to deal with that. And it means that if you want to deal with flexibility, that effectively you have to, the docking problem becomes harder, because instead of having only a 6-dimensional search problem, 3 rotation, 3 translation, you need also to sample rotations around bonds in your molecule. And the scoring also becomes typically more complicated. One big problem and challenge in the field is it's difficult to predict a priori conformational changes. Well, actually, you can predict conformational changes. You can use molecular dynamic simulation, you can use elastic network models, but what is difficult to do is to know when you need those and when you do not. In general, in modeling, my method will be start with the simplest approach possible, and only if you have good reasons to go to very sophisticated approaches, go to that. Because in modeling, in docking, if you do very fancy, enhanced sampling, molecular dynamics, take snapshots, and if you don't need those conformational changes for the specific binding process you are interested in, the results at the end of the day will most likely be worse than if you had not done all those fancy sampling techniques. So if we were to know when do we need to sample conformational changes and when not, the problem will be much easier. But it's simply difficult to know beforehand when things are going to change. Sometimes experimental methods might tell you that something is happening. And because of that, pretty much all the docking methods out there can only deal with rather limited conformational changes. We use benchmark to measure how good we are doing in the field. And for protein docking, we have a benchmark of about 250 complexes. And they are classified as easy, medium, and challenging. And the challenging category starts already with conformational changes as small as 2.5 angstrom root mean square deviation. Okay, so what we cannot do is fold a protein which binds to its partner. So binding-induced folding is not something we can really deal with. So we are really limited in what we can do. Also, you have to realize that the treatment of flexibility will depend on the method that you use to do the search. So if you use grid, a grid will be rigid by definition. So you will have to handle flexibility in different ways. So one of the first way of dealing with flexibility is to do what we call soft docking. So what we are doing is basically to kind of mimic small conformational changes. We're going to allow overlap between atoms. So this will be hard sphere, no overlap. This is soft, rigid docking, overlap. So it's an implicit way of treating flexibility, and you will need to refine the solutions because they will have clashes. So this is what's happening also typically in like grid-based docking methods. So this soft docking in grid-based method can be implemented, for example, by emptying the grid for the side chain on the surface of your protein. So you still have the representation of your surface, the blue voxels here, and then you empty the grid points that correspond to the side chain or the region that you might want to make implicitly flexible. And it means that if you have overlap in this region, it will not penalize you. Because if you have overlap on the gray square here, this will give you a penalty in terms of energy. But you will have to refine those models. What you can also do is, instead of starting from a single structure, is try to pre-sample structures before doing the modeling, before the docking, and use an ensemble of conformation as starting point for the docking. Now, depending on the software that you are using, you can give the ensemble of conformation to the software, or you will have to repeat the docking for each conformation that you are sampling. Okay, so methods, grid-based methods, you will have to repeat the docking for each conformation. And here the sampling can be done again by enhanced techniques like molecular dynamics, enhanced sampling, elastic network models. You might have in a PDB different conformations of your protein. So there are different ways of generating those. And this is applicable both for rigid and flexible docking. And then comes the methods that can explicitly describe flexibility during the docking and during usually the refinement process of the docking. And here you might introduce rotations around side chain and backbone. So this increases the computational cost because you have to do more work, which is why it's usually introduced later on in the refinement process. And then finally, the scoring aspect. So we're going to generate tons of models, and then we have to select what are the good ones and distinguish the good ones from the bad ones. And this is really the holy grail in docking. If we had the perfect scoring function, the remaining will be computing time. Okay, and then we have beautiful HPC center where you might get access to a lot of computing time, and then you could solve that problem. But the problem is that we don't have the perfect scoring function. And how you score depends also on how you represent the system and how you deal with flexibility. And what you see in the field is that there are scoring functions that are made very specific to specific types of problems. Protein small molecule, antibody antigen, enzyme inhibitors. So, but in nature, in principle, the physics, the chemistry, the rules are the same. So what governs the binding is the free energy of binding, gives free energy. But the scoring functions are not giving you a free energy of binding, not a binding affinity, they are giving you some kind of score. So what do you find in those scoring functions? Usually, there are some kind of linear combination of different empirical terms, such as intermolecular van der Waals energies, electrostatic. You might look for hydrogen bonds across the interface, the amount of surface which is buried, dissolution energy, removing of water from the interface. You might try to calculate some entropy loss based on the number of side chains that are buried. You might use statistical potentials derived from analysis of non-structure to score. And you might even use experimental data, if you have those, to score your problems. So many scoring terms are based on the surface accessibility. So the solvent accessible surface area, STASA, where you basically can calculate that. So the original paper dates from 85. So you can calculate the amount of surface which is accessible to the solvent. And when you form a complex, what is interesting to look at is the buried surface area, which is the difference between the surface area of the separated molecule, so molecule 1 plus 2, minus the surface area of the complex. And typical values for protein-protein complex are between 1200 and say 2200. This is where you find the peak of the distribution. But of course, there are complexes that are very small interfaces and are very strong binders. And you will also find complexes that are very large interfaces. The dissolution energy, which I mentioned, is also related to the surface area, but it's an empirical term where you basically now calculate the surface area per atom, so it's an atomistic calculation, and you have a parameter which depends on the atom type. So, and then you calculate again the solvation energy of the free molecule minus the solvation energy of the complex, and this is giving you the dissolution energy, the removal of water. So if you buried hydrophobic surfaces, typically it's good, it's a bonus in energy. If you bury charges, usually you have to pay a price in energy because you have to dissolvate those charges. Here is an example, old example of statistical potential. So this was published in 1999. There were not so many complexes in the PDB at the time, and there are still not so many complexes in the PDB today. We estimate that the number of complexes in the PDB today, biological complexes and say unique, non-redundant entries of biological protein-protein complexes is in the order of 7,500. Okay, if you search, give me all the entries in the PDB that have more than 2 chains, you're going to find a very large number. But only a subset of those are real biological assemblies. So back in 1999, they did statistics on the PDB, and you can define some kind of potential. If you find a contact between 2 aspartate, is it good or bad? No, most likely it's bad because they are both charged. But you can use this kind of statistical map, and since then there are more atomistic description of those to score your complexes, and it's implemented in various software. And what is also often done to simplify the scoring is to apply clustering methods to the solutions that are generated, where instead of having to look at thousands of tens of thousands of models, you're going to cluster them based on different metrics, which could be RMSDs, but which could also be the fraction of contacts. So how many of the contacts across the interface are similar? And then instead of looking at thousands or tens of thousands, you might end up looking at a few tens of clusters. And this simplifies the scoring problem. Are there any questions, additional questions at this time? If not, we are going to move now into more the specifics of ad hoc and information-driven docking. So until now, we have been dealing with docking, general aspects of docking, and now we're going to speak really more about how we are doing this kind of modeling using ad hoc and using information. Now, ad hoc has been developed for almost 20 years now, starting from NMR data actually to guide the process, but since then we have been adding support for many different kinds of data. And you will see in the tutorial, we are actually going to make use of this kind of NMR titration data, but I already explained to you that there are many different types of experimental and bioinformatic sources that can be used to guide the modeling process. So ad hoc will allow for us. So it's not, it's using an explicit representation of the system. We are kind of energy driven. So we use forces to drive the optimization process. What distinguishes ad hoc from many of the docking software is that we can model more than 2 molecules at the same time. All the grid-based approach, for example, can only handle 2 molecules and not more than 2. But we can currently go up to 20 molecules, but it only really makes sense to model a large number of molecules if you have good data. Otherwise, it's simply a waste of computational resources. I should also mention that symmetry is also a source of information. So if you know that you're dealing with symmetrical complex, you can use this information to bias the modeling. And we have been consistently participating to Capri over the years, and we have shown that using, in this case, mainly bioinformatic predictions and maybe mutations, we can get very good performance. So how do we encode the information in ad hoc into some kind of energy functions that's going to drive the docking process? So the main concept in haddock is the use of ambiguous interaction restraints, or what we will call also ambiguous distances. This is not new. This was introduced in NMR structure calculations by Michael Nilgius and Axel Brunger back in the early 90s. And what you see here is an energy function that represents actually the NMR distance, NOE distances from NMR, which has, so you have an upper limit and lower limit to your distance. If you are in between those two, the energy is zero. So you see the energy function here. If you are below the lower limit, the energy goes harmonically higher. So this is this part of the energy function. If you are above the upper limit, the energy is also, you also get into the harmonic part of the function. But the problem with the harmonic function is that it becomes very, very large very quickly. And this will cause instabilities in the computation. So for NMR, what basically they introduce is another type of function which becomes linear. So you have an harmonic term here, and then it transitions to a linear energy function. Now, the energy here still goes up. So it means that if you have very large distance, you will still have a high energy. But since we are using here gradient-driven optimization techniques where we calculate the forces, and the forces is what is important here, it's the slope of this function basically which is important. And once we are in this linear region here, the slope becomes a constant. So the force is no longer increasing as you increase the distance. And this means that you can have stable computations when you are in that regime. So now, how do you encode information about, say, knowledge of residues that are at the interface, but without knowing what are the contacts that those residues are making? So what we're going to calculate is an effective distance between a residue that we identified, for example, from mutagenesis, and the entire interface identified or predicted on the other side. And we calculate all possible combination of atom-atom distance between these residues and all residue part of the interface on the other side. All those distances we're going to sum as 1 over the distance to the 6th power. This is dipole-dipole interactions. So this is also coming from NMR actually, but this is also the attractive part of a Lennard-Jones potential to represent van der Waals interaction. So this sum, as you add more distances, this sum is increasing and increasing and increasing, and then you take the inverse of the sum and the 6th root of the sum, and this gives you 1 distance. So for each residue here that we define as being part of the interface, we will have 1 distance restraint during the optimization process, but it's going to be defined to the entire interface on the other side. This distance is always shorter than the shortest distance that enters the sum. So this is a property of this summation here. So this is the way that we can encode basically a distance restraint without knowing what are the exact pairs of atoms that are should be in contact. Now, there was a question about the bias. If you use data, how can you minimize the bias? So you have to trust your data. But the problem is there will always be, the data are never perfect. So there are several ways, but one way of solving the problem that not all the data will be perfect is this part of this energy function. So here the force becomes constant. So a violation of 100 angstrom or a violation of 10 angstrom is not going to give a different impact on the energy optimization. So that's already one way of minimizing the bias. And the other way is that we randomly, by default in haddock, we randomly delete 50% of the information that we have. And we calculate the models with the other 50%. And you repeat that for every model that you generate, meaning that from time to time, you're going to throw away good information and get bad models, but then you also from time to time throw away bad information and get good models. And then it will be up to the scoring to distinguish what is good from bad. Now, we have to search this interaction space. So we use now a classical force field as is used in molecular dynamics, for example, where we represent bond angles, rotation around bonds, and we have the non-bonded interactions, very important for molecular recognition, and the experimental information. So this can be this distance term, but we also have all type of energy functions, more NMR related, but we also have, for example, cryo-EM supporters. And the search in haddock is a combination of energy minimization and molecular dynamic simulation. So we use gradient-driven optimization techniques. The protocol itself has 3 stages. In the initial stage, we're going to treat the molecule as rigid, rock solid, and we're going to use energy minimization to bring the molecule together, driven by the data that we put in. HADOC also has some ab initio modes. I might tell you about that during the tutorial. At this stage, we're going to generate 10 or 100,000 models, and we write a fraction of those to disk. Then we do a first scoring, and then we optimize the interface. So here we introduce flexibility into the process to optimize the models. And at the end, we might add a little bit of water on the protein and optimize again. This is nothing close to stay a proper molecular dynamics simulation. It's only a very short optimization. In the latest version of HADOC, we don't even do that anymore. We only minimize. So you see here the minimization problem. So you see the molecule are first separated in space, randomly rotated. So these random rotations are repeated for each starting point for the minimization, so that we sample different starting configuration. And in this case, you see the spheres represent the experimental information. So here we had NMR data to drive the modeling process. So you see the molecule rotating, translating to form the complex. We then take the best ranked model at this stage, and then we start doing now molecular dynamics, actually in torsion angle space, first optimizing the side chain at the interface, and in the second phase, the backbone as well. If you look at this loop, it just flipped over. So in this way, we can describe, say, small conformational changes that are happening upon binding. And by small, it can be 1 angstrom, it can be 2 angstrom. Sometimes we go up to 5 angstrom, but only in cases where we have a lot of good experimental data to drive the process. Otherwise, don't expect miracles. And this will be the short molecular dynamics. And short, we're speaking of picoseconds here, a few tenths of picoseconds. So this is nothing like a proper molecular dynamics. Mainly, we are only optimizing slightly the side chain. Nothing changes in the backbone anymore at this stage. And the scoring gets slightly better. But this stage is not even done anymore in HADOC by default. So flexibility, I already mentioned several times. This is a big challenge in the field. So how can we deal with flexibility in HADOC? So we can provide an ensemble of conformation as starting point for the modeling. And then HADOC will take all possible combinations from those ensemble as starting points. And we also use the scaling down of intermolecular interactions during the simulated annealing optimization process. And we also have this explicit refinement where we allow for rotations on backbone and side chain. Now, something about scoring. So we have 3 main stages, and we have different scoring functions at the different stages. This is the final scoring function where we have 20% of the electrostatic, intermolecular interactions, intermolecular van der Waals energy, the dissolvation energy term, and the term that will represent the experimental information. So this was optimized on a few complex almost 20 years ago, and it's still surviving a lot of optimization. You can always make it slightly better, but you will make slightly better for protein-protein, but then it will not work optimally for protein peptide or protein nucleic acid. And we use function pretty much for all kinds of molecules that HADOC support. So we can handle proteins, nucleic acids, both DNA, RNA, peptides, small ligands, glycans, and for in most cases, we use the same scoring function, which is basically measuring the amount of energy between the molecule, accounting for the dissolution of the interface, and accounting for the restraints that are defined to guide the modeling process. Questions? Let's see some.
We have a question here about how does HADOC perform against other methods?
So I guess HADOC is quite, it has first a number of unique properties in the number of molecules that you can model, in the information that you can input. So I think most of our users are using some kind of data to drive the modeling, which might not be possible in other software. Now, if you have no information at all, so we have an ab initio mode, but I think if you're only dealing with 2 molecules, there are other software that might be better suited for the problem. Like Plus Pro is doing very well in Capri. So if you want to compare things in, say, in a blind mode, I would recommend you to look at the protein papers, the protein issues dedicated to Capri. So over the years, we have been competitive in Capri, doing well. You lose some, you win some. So it all depends. if you have data to drive the modeling process, I would say that we are doing quite well compared to other software. So it depends on the scenario. It's, as you will see, it's being used a lot. So I think usage and citation that we are getting is very high. So this is probably demonstrating that we are doing something useful. But again, you know, there are different software out there, and some software might be better suited for your specific problem. Anything else, Rodrigo?
That's it.
Okay. So most of our users are actually using the web portal implementation of Haddock. You can also run it locally, but the portal does much more work for you in terms of pre-processing and post-processing, as you will see. So we have more than actually, I think 23,000 registered users these days. We have served almost 350 docking runs since the server has been up and running. And we can do that because we have access to high throughput compute resources, not per se high performance compute resources, but I'm going to come back to that a little bit. You can also get the software to run it locally. So this is a world map of our user base. Most of you are based in this region of the world, attending the course. I just checked. We have more than 350 registered users in Malaysia. I think about in Thailand around 150. And then Vietnam, I saw a lot of you from Vietnam. Vietnam is less represented, about 50. But there's already quite a number of users in the region. India a lot, and then you have Europe aggregated and the US. And it's being used a lot. So this is a plot showing you the number of submissions to the portal, the number of docking run processed per month since about 2 years, and number of single users active per month on the portal. And you see, before COVID, we were about 300 users per month that were active and about 3,000 docking runs per month processed. And then comes COVID and the lockdown, and you see that we are actually seeing the COVID waves in the usage of the portal. So we doubled the number of users, even triple almost, and we more than almost tripled the usage of the portal. And since April, we're also monitoring the jobs that are related to COVID. So you see about 1/3 of all the submissions to the portal since then are dedicated to COVID-related research. We can provide those resources because we have access to a distributed resource, high throughput compute resource provided by EGI, the European Open Science Cloud, and also the US Open Science Grid. And there are also sites in Asia actually that are supporting our computing, in China and Taiwan especially. And in the past, we were also computing in Malaysia actually. Well, you can find this kind of, so by the way, Haddock, it means fish, of course, in English, but there's also a comic figure, very famous, at least in Europe, which is Captain Haddock. It's here, hidden in this suit. And this is Tintin, who is the big friend of Haddock. And this is actually a hybrid representation because this was the album Tintin Going to the Moon, and then the Blue Lotus, which happens in China. They made kind of the adventures of Tintin and the coronavirus out of it. Now, computational channels, to put it in the perspective of HPC computing. Let me check the time, yes. So the server for sure is running in high throughput compute mode. So we are sending per year more than 20 million jobs to the HTC resources that are distributed around the world. But we also, you can also run Haddock locally on your HPC resources. And within the BioXcel project, which is a European project, Center of Excellence project, we are also preparing Haddock to run at exascale on the new HPC center. And you see here an example of so a different way of running Haddock, so not through the server, but running locally on HPC resources, where you see basically this is wall clock time. So these are minutes to calculate 100 complexes, and as a function of the number of cores that you are using, actually the number of nodes. Okay, so these are 40 or 96 core nodes that we are using in these. And it's pretty much a linear scaling that you are seeing here. So we hope soon to be able to benchmark also on a much larger scale in a pre-exascale system in Europe. Now, if you were to, in the title I mentioned interactome, but let's think of the human interactome. So in our proteome, we have about 20,000 proteins, okay, ballpark numbers. Those will form more than 400,000 interactions. So the number of complexes is way larger than the number of players. Now, if you want to model all of those, okay, we will need more than 10 million CPU hours if we use our software. Some of our software might be faster, of course. And I think another big problem, so CPU is not only the issue here, but if you want to go to large scale modeling of interactions, the other problem that you have is that you're going to generate huge amount of data. When you do docking, you generate a lot of files typically, which is very different than running a molecular dynamics where you generate one large trajectory. And this causes challenges to the file system on the HPC resources. So this is something that we will have to address as well. So not only the CPU and the scaling of the computations, but dealing with the amount of data that we are generating. And we are doing that in the context of the BioXcel Center of Excellence. And under BioXcel, we are also operating the Haddock Forum, where you can find a lot of, when you can ask questions actually about Haddock and find answers as well. So often the questions that people are asking in the forum have already been answered. So the answer is somewhere in there. So this is our help center. Next to Haddock, which is our flagship software, we are operating a number of other portals and software in Utrecht. A lot of those are related to interactions. Prodigy and Spot-on are related to binding affinity and hotspot prediction. DSVs to visualize the amount of information in distance restraints between complexes. PowerFit to fit rigid models into cryo-EM map one at a time. These are bioinformatic prediction methods to predict interface, which might feed into Haddock. And we also have something dedicated to predicting the binding site of the paratope in antibody based simply on sequence. Any more questions at this time? So we have about 15 minutes to talk, so I will just move to show you a few examples and some highlights, and then we can move to the tutorial. So one example of the kind of modeling that you can do with Haddock is this complex, which is a membrane protein, FUS-A. This is actually it sits in the membrane of the bacteria, and this bacteria is targeting plants. Okay, so and for its survival, the bacteria needs iron, and it's going to hijack iron, hence the piracy here, by binding to a small protein of the host, ferredoxin, which contains an iron sulfur complex. And here you see Captain Haddock again as a pirate this case. So in this group of researchers that are distributed around the world, they are crystallographers, they are animal people, and they are modelers like us. So when doing the crystallography of this system, they never managed to co-crystallize the complex. So they could crystallize the membrane protein, which is a tour de force, and the membrane proteins also, without even knowing the complex, we do have information because we know which regions are in the membrane. So the binding must happen in this region, which is the extracellular region. So this is also information in principle. Now for ferredoxin, what was done is to use NMR to map the binding site on ferredoxin. So by using titration experiments and looking at displacement of signals, you can basically plot the displacement of signal as function of the amino acid sequence here on the x-axis, and you see that there are specific regions along the sequence that are affected by binding. And if you visualize those on the surface of the protein, this defines a well-defined binding patch. And you see here in yellow the iron-sulfur cluster, actually. Now, this is information that you can use in Haddock to define these active residues. For the membrane protein, we cannot do the same experiment, but we know that the binding has to happen in the extracellular loop. So we're going to define those extracellular loop as the binding region for ferredoxin. And we give this information to Haddock to drive the modeling process, so we don't need to sample the entire space, but we target the docking. And this is the top cluster coming out of this modeling process, which of course will need to be validated by mutagenesis. And this was not yet done when the paper was published. Now, some recent developments I already mentioned, we can go up to 20 molecules of different types, so you can mix and match molecule in Haddock. And if you want to read details, I refer you to this Nature Method paper, 2017. We have added support for cryo-electron microscopy data. So we have an energy function that represents the data, and you can dock into the EM using distance restraints first to get the molecule inside the density. And once the molecule are inside the density, we turn on the energy term that described the EM data and refine against that. This was described in 2015 already, but implemented in a server only rather recently. We have also, in order to go to larger system, more complex system, we have built a coarse-grained force field into ad hoc using the Martini force fields, both for nucleic acids and proteins. So these are described in those two papers here. And where basically we are transforming an atomistic system into a coarse-grained system. Here we replace 4 heavy atoms by 1 particle, so you have less coordinates to deal with, which simplify the search and speed up the search. We do the docking at the coarse-grained level, and the final stage of ad hoc will be to transform back this coarse-grained representation into a fully atomistic representation. And the server supports this simply by switching a switch. If you want to, compare the performance of ad hoc with respect to other software, you can look at this paper published last year about modeling antibody-antigen complexes. And we compare here the performance to other software like LiDoc, ZDoc, and PlusPro. All the other free software do allow to use information, knowledge of the binding site, the hypervariable loops on the antibody to filter solutions. So they have been optimized for antibody-antigen docking, so you can compare how well ad hoc is doing with respect to that. Since we use the data directly to guide the modeling, we are actually doing better than the other software in general. And a very recent story, which is available on bioRxiv, is work we have been doing on small molecule docking, and this is a new protocol where we basically do a kind of template-based docking, identifying a template which contains a ligand most similar to the ligand that you have to dock, transforming this ligand into some kind of shape information, and defining distance restraints between the ligand that you want to dock and this shape to guide the modeling process. And this is giving a very competitive performance to all the commercial software out there. So if you want to know the detail, look up the paper on bioRxiv. So time to conclude and finish to give.
So we have a quick question before we finish. I think it relates to the last slide that you just showed about the small molecule. And the question is, how does ad hoc identify the binding pocket of a target protein?
So basically, that's something that you will have to, you could do an ab initio docking. One of the tutorials that we have online is about that. But there's pretty much all small molecule docking software are going to need some definition of the binding site. If you use Autodoc, you have to define a box around your binding site. So there is no difference in ad hoc than other software. If you have no information at all, you could try to dock against the entire protein and then identify possible binding sites. So there are also software like F-pockets that allow you to identify binding pockets, and then you will target the docking to those regions. But this is very common to all docking software. So in conclusion, and it's just one slide of conclusions, I hope to have convinced you that integrative modeling, information-driven docking is useful to generate model of complexes, and even when you have based on information. You have to realize that what you're generating are models, okay? This is not the truth, this is not experimental data, but the models are very useful because they provide you a starting point to generate new hypotheses, which you can go test experimentally. So modeling and integrative modeling is very much complementary to the experimental methods. So a big thank to my current group. And this is how we are holding our group meetings. And this is Rodrigo, you might have spotted in a Zoom meeting. We can do everything that we can do because we are getting some funding over the years from the Dutch National Research Council, but also from the European Union, very important for all the work that we are doing, especially BioXL. And thank you all for your attention. And if additional questions, we can handle them now.
I'm not sure if you're checking the chat, but there is a new question that popped here. Is for the latest protocol that you just showed in bioRxiv, So how does it deal with alternate conformations of the side chains in the active side?
The active side. So you will have flexibility during the docking process in Haddock. So we allow for flexibility. That's the standard protocol. You could give an ensemble of conformation to Haddock as well. But I think the critical issue in the protocol, and again, look up the paper, is to identify the template which has the ligand most similar to the ligand that you need to dock. And depending on the similarity between ligands, you can expect a very good docking performance or a poor docking performance. So it's all documented in the paper. So I don't think we have time to go into the details there. But I think for small molecule docking, really the choice of the receptor is critical. For the ligand, it's less of an issue. We generate those from small strings. So we generate an ensemble of conformation. And because we are giving restraints to the shape, we can induce quite large conformational changes in the ligand during the docking. So that's the advantage of using the restraints to guide the docking process. Spot on, I also see. So spot on, that's the model where we use different, so energetic, buried surface area, sound accessibility, conservation. So there, and it's a machine learning model to identify hotspots. So it has been trained to distinguish hotspots from null spots. So you can again look up the paper if you want to know all the details. Any more urgent questions? If not, then we can, so we have a small hours to go into the tutorial part of today and start using Haddock. So I'm going to first paste again the location of the tutorial in the chat. And I'm going then after that to share my screen and guide you through that. Okay, so you should be seeing my screen. And this away for now. So the link to the tutorial that we are going to do is in the chat. And otherwise, you can also find it actually if you go to bonvinlab.org. This is the group page of the web page of my group, and you find here under education, you find links to tutorials. You also find links to a number of lectures actually that have been recorded in other workshops, so you can watch back those next to the lecture I gave you today, but there is probably even more content in your lecture. So here you have a number of different lectures. And I think very important, if you're going to use Haddock, before we go into the tutorial, we have this best practice guide. So I mentioned we can do a lot of different types of molecule, and depending on the molecule type that you're going to dock, there might be settings that you might have to change. The server will change those settings automatically if it detects the right molecule, if you provide the right molecule, but if you run locally, you will have to do that yourself. So really, read the best practice guide before starting, not today. You also have to worry about how to prepare the structure for Haddock, because they're like, how do you give an ensemble? But you also sometimes there are issues with crystal structure that might have multiple conformation of the side chain. So we need to deal with that before the docking. So take time to visit this best practice guide if you are going to use Haddock in your work. And for today, we're going to the Haddock version 2.4 tutorial. We still have an old version of the portal running, but this will be discontinued probably at the end of this year. And there are some other tutorials then. And under Haddock 2.4, we have quite a number of tutorials. Some are related to the use of cross-link data from mass spectrometry, multi-body docking, using DSVs, for example, kind of a template-based docking protocol. ab initio without information for symmetrical tetramer. Ligand binding sites, this is not yet the shape protocol. Oops. Something here needs to be corrected, so that's a mistake. So the shape protocol will appear soon here, okay? But the ligand binding site, if you have no knowledge of the binding site, this will be the protocol to look. But for today, we go into the basic one. Now, all our protocols on the page are set up in a similar way. So what we are going to do in this particular case, we're going to use NMR data that are defining the binding site of those two proteins to guide the modeling process. And we're going to set up the docking through the web portal. Again, here there is, so in all the protocols, all the tutorials we have this color scheme. So if you see orange, this is a question, so we should think about it and try to answer it. Blue will be an instruction, you need to do something. So instructions will be used, for example, to enter data through the web portal. Green will be a PyMol command in this particular case. It could be also camera in different protocols. And some protocols, some tutorials will have black as well. And black will be Linux level command line commands that you would have to do. So you should have registered to use the portal already as instructed. Again, some information about haddock. You can see here again the small optimization movie that I show you during my talk. But now we can go directly to the core of the tutorial. So first thing we need to do, and this is true basically anytime you do docking, you always have to inspect the proteins that you're going to dock and clean them because they might have all kind of stuff. So we're going to use PyMol for that. So these green commands are all PyMol commands. So I'm going to start PyMol. Okay. So PyMol, if you are familiar with PyMol, then you can probably proceed on your own, but PyMol has different ways. So there are menus here and there are menus on the, so basically up there you can use, but you can also type commands in PyMol in two locations. Up here, there's a prompt, and at the bottom as well. You can copy, paste commands from the website. So I'm going to copy this first. So what this is going to do is to fetch a PDB file from the PDB directly and display the molecule as a cartoon. And it's going to also show me the histidines in this case. And if you want to paste command into the command line of PyMol, you have to do that in the top window. It's not working at the bottom level. So in the top window, you can copy paste. And this is what you are seeing here. Why do I display the histidines? If you read the information about this tutorial, this complex, so the biological function of this complex is to transfer a phosphate group from a histidine of one protein, from E to A, this protein, to a histidine of HPR, another protein. So that's the biological function, which is why we want to look at the histidine. And the first question comes, are there any histidines in this system? Well, there are histidines, yes, we see two here, but you see there is no phosphate group present. Now, what can be the charge state of histidines? Question to you. Anyone wants to say something or put in a chat? Histidine residues. Positive charge, they can be positive or neutral. Each IP, so that's, the naming is very important. So we are following PDB namings. So if you use Amber, for example, they use different namings. So histidine can be charged or neutral, depending on the pH. And if we add a phosphate to histidine, it's going to change completely the charge state because a phosphate will carry a negative charge. Okay, so if we want to do proper docking, we should represent the phosphate. So we'll go to that. You also see crosses here around the protein. What are those? So this is a structure I downloaded from the PDB. This is water. Okay, so it's a crystal structure. You see water molecule. In this case, there's only water there, but in some cases, you might have crystallization buffer coming up also in your crystal structure. So you might have, small molecule co-crystallized, which you probably don't want if you want to do docking process. Okay, so if you don't remove those molecules ad hoc, at least the server will keep them, and that might give you problems. So it's always good to really look at your files when you prepare your docking runs, remove anything that you don't need. Also, if you know that the binding site is in a specific domain of a very large proteins, you might not need the other domain of that protein. So simplifying the system will lead to a faster docking. So there is no phosphate, we answered that question. So we can remove the water, so you can do that in Pymol by simply saying remove water. And if there was other molecule, you could remove them as well. Now we can see what NMR is telling us in terms of binding site. Okay, so these residues that you see in blue here are the ones that have been identified. If you look at this paper, EMBO Journal 2000, they give a list of residues affected by the binding. So let's visualize that. So we're going to show a surface representation of the protein and color in red the residue which have been identified by NMR as being affected by the binding. Yep, copy paste. Okay, so you see now a surface representation of the protein, which is also shown in the tutorial. So the first question is, does the residues identified by NMR form a well-defined patch? Now, if you rotate the molecule, you see on the backside there is nothing, so that's good. And on this region, you have kind of a donut shape here. So you see that NMR is not detecting, so it will be very surprising if the residue here in the center of the donut are not involved in binding as well. But you see that NMR experimentally is not detecting the full surface, okay? So it's not a contiguous surface. So it's a well-defined patch, but it's not a contiguous surface. So for docking purposes, what ad hoc is doing in the concept of ad hoc is that we're going to define the surface neighbors of those red residues as passive residues. In the way we define restraints in ad hoc, the red residue should be making contact with the other molecule, otherwise it will be an energetic penalty. But the passive residues, that will be the surface neighbors, they can make contact. But if they do not make contact, it's not penalizing you in terms of energy. So now that we have the molecule ready for docking, we have removed the water, what we want to do is save it to disk so that we have it ready for ad hoc. So I'm going to export the molecule. I'm going to save it, and I want to save it. I'm saving it in a tutorial directory, and I want to save it in a format, in a PDB format. So there are different formats that you can use in Pymol, so you want to use the PDB. In general, it's better to get rid of the water because, if the water is in crystal structure, water will be all around the protein, and it will prevent basically the binding to occur properly if you don't need it. So always start simple. So if you want to keep water, there should be a good reason why you want to keep water. Otherwise, simplify your system. So I'm saving it to disk. Okay, and now I'm cleaning, delete everything. So now we have our first molecule ready. So now let's look at the HPR structure, which is the binding part. So I'm going to fetch it again from the PDB. Okay. So this is, so there are no water molecule in this case. Let's look at the binding region. You might notice my computer takes a bit more time to generate the surface, which is strange because it's a smaller protein than the other one. Now, if you look at things, again, the NMR is pointing to a very specific region of the surface, and it's doing quite a nice job at pinpointing the binding region. So that's nice. There is no water in this structure for the good reason that this is actually an NMR structure. So if you download NMR structure from the PDB, what you will find usually in a coordinate file is not a single structure, but an ensemble of structure. So let's visualize this ensemble. So what I'm going to do here, I'm going to display in a ribbon the representation in PyMol the ensemble, which basically only gives you the C alpha carbon trace. And now you see that this is not a single model, but it consists of actually 30 models. And in the PDB file, if you open the PDB file as a text file, you will see there is statements like model 1 and then and model. And then you get model 2 and model. And you see that there is some conformational variability in those models. And you still see the regions that are detected as being part of the interface. And you see that in some of those regions, there are small changes in the backbone. So it could well be that one of these conformations is better for the binding than another one. Okay. But here we're only looking at the backbone. So let's visualize the side chains, but only of the residues, the red residues that have been identified by NMR. And we have defined those as a selection HPR active. And now you see here that there is much more variation at the side chain level than there is a backbone. So here you see one specific amino acid, which has a huge variation in the orientation of this side chain. It's quite normal in NMR because typically you have less information to define the exposed side chain. So it's different from crystallography. Which amino acid is this? Question to you. Tyrosine, there is a tyrosine here, but that's not a tyrosine. Tyrosine will have an aromatic ring. So there is something here that looks like a tyrosine, but this one is actually a phenylalanine because it doesn't carry the, there is no oxygen attached to it. So that's not a tyrosine, this one. To help you, it's a charge residue. And if you know PyMol, you just click on the side chain, it will tell you what it is. Arginine, yes. So this is an arginine. So arginine is positively charged. So that's a residue which is actually quite important for interactions. We do find arginine quite often at the interface of complexes, while we do not find lysine residues very often. So it has a softer charge. And you see, so this side chain here is going from this side to the other side. So it could well be that one of these conformations is better for binding than another one. These kind of conformational changes will be very hard to model during the docking process, okay? So you need to switch from one side to the other. Once you have generated the docking pose with the molecule docked onto each other, these kind of conformational changes are not very likely to happen. So if we use an ensemble in this case, maybe we have a better chance of getting a good docking solution than if we were to select the first model. So we're going actually to use the ensemble of conformations for the docking process. So same as before, we want to save this. So export molecule. We're going to save all states, and you want to save it again as a PDB file, and you might call it HPR ensemble.pdb, or whatever you prefer. Save. Okay. So in principle, now we are done with preparation of molecule. I'm going to close PyMol for now. Going to the command line. You don't need to go to the command line, but so if I look, what do I have in my directory? Increase a bit font size to make things more readable for you. So I have my two PDB files. And now we have to worry about the phosphate group. So we basically need to tell Haddock that there should be a phosphate on a specific histidine. And the histidine that we need to look at is histidine number 90. So we know that. Now in Haddock, we have support for a number of modified amino acids. And you see here on the tutorial page, there's a link to this specific page. And in this page, you will find all the modified amino acids that are supported by Haddock with their name. So I'm going to open that in a new tab. So you see, for example, supported carbohydrates. Okay, so you have carbohydrates, you want to dock carbohydrates, or you have a glycosylated protein. Those carbohydrates will be recognized by Haddock. We support ions as well, but ions should be entered in a very specific manner that contains the charge state information. So you will have to edit your files to that. Some cofactors that are supported. So the naming is important here. Nucleic acid, bases, DNA, RNA, how do we name them? And then you get the list of modified amino acid recognized by Haddock. So it's quite a number that we have that we support, but again, the naming is important. The naming of the atom usually is rather standard in PDB files. What Haddock will do as one of the first stage is to build all the missing atoms. We don't need to worry about missing atom. We can let Haddock build those, but we need to worry about the naming of the residues. Now, if you were to search here, you find here NEP, which means it's a phosphorylated histidine where the phosphate is attached to the N epsilon atom on the aromatic ring of the histidine. Okay, so we need now to edit the PDB file, change the residue name of residue number 90 to NEP. And this is all we need to do for Haddock to recognize this as a phosphorylated histidine. And it will, during the docking process or before the docking process, actually generate the phosphate. So you can use here any text editor. So it should be an ASCII text editor. So don't use Word to open PDB files. It's not a good idea. But Notepad, if you're working on Windows, I'm going to use VI at the Linux level, command level. If you don't know that, don't start doing that. I need to search for residue number 90, and here I find it. Okay, so we have here histidine number 90, going back to the tutorial. Okay, so you need to edit the PDB file that you save in your favorite ASCII text file editor. So you need to save it as a simple text file, ASCII file, and we need to change the name of histidine 90 to NEP. Okay, so I'm going to do that. And that's it. Now I'm going to save that file, and you can save it under the same name, or you can give it another name just to distinguish between the two. I'm going to add the P to the name. And that's all I did. So I did not add the atoms, I just simply edited the name of the residue in the file. And I can show you the difference between those two files is simply indeed in histidine 90. In one case it's called his, in the other case it's called NEP. The coordinates are exactly the same. This is, by the way, the inside of the PDB files. Often in workshops there are people who have never realized that the PDB files are a simple text file. I hope it's not your case, but it can happen because you are used to opening them in graphical display like Bimol or other software. So now if you have done those steps, in principle, you are ready to submit. It's important that you don't forget to change one atom to the proper naming, because otherwise Haddock will complain that there are two residues, number 90, that are different, or two residues, number 90, which are different amino acids, and that's not going to work. So now we are ready to set up the docking run. So we're going to make use of the 2.4 interface of Haddock. We can go through the steps. You don't need to submit those calculations. You can, but it's not needed because we have pre-calculated data, okay? But we'll just go through the process. So I already have the server here, no, not that one. Okay, so let's go to that one, open link in a new tab. So I'm already logged in this case. So if you are not logged in, you should log in. On the page will ask you to log in with your credentials. So you should give a name to your run. So this is E2A P-HPR. You can see here now the number of molecules. So if you look at this form, by the way, all these blue menus, you see an arrow, so you can close and open those menus. So this will be the first page of the server. If I was to change the number of molecules, the menus will be automatically updated. So you can see we can go up to 20 molecules here, but for now, it's only 2 in this particular example. And we are going to submit it. You could give a PDB ID and download it directly, but this is not a good idea because, again, I show you that there is usually a lot of things in those PDB that you don't want to keep for the docking process. So we're going to submit it ourselves. We're going to use all chains. And then I'm choosing the file, and the file is the E2AP in that case. So the one where I changed the naming to NEP. Upload. The default is protein or protein containing ligands, but you can find here the other type of molecule that are supported: small molecule, peptide, glycans, nucleic acids, protein nucleic acids. Like if you were to use a nucleosome, which contains both nucleic acids and protein in the same molecule, this is what you will have to do. And you'll see this shape here, which is what you will need to use if you were to set up the shape small molecule docking. And if you are patient, there will be a tutorial describing that soon. I mentioned coarse graining. You could coarse grain your model at this stage, but then there is the phosphate will not be supported at the coarse grain level. We only have the standard amino acid supported there. Other options, important thing to realize is that the termini of the molecules are by default in ad hoc, uncharged. And there's a good reason why we do that, because often what you get from the PDB in terms of crystal structure is not the full sequence that has been used in the experiments. If the termini of your proteins are disordered, there will be no density for that in the crystallographic data, and you will not model it. Meaning that what you have in a PDB file is not the real end of the molecule, but there is a peptide attached to it. And as such, it should not be charged. Okay, so think about the termini of your protein, because again, if you add a charge or not, it might make a big difference in the docking results. Sometimes, doing a mutation is sufficient to kill a complex. Now, adding a charge might have the same effect. So by default, they are uncharged, but if you want charged termini, you can change those. Note that the options that you can control on the server depends on your access level. Okay, so I have Guru access, so I can change anything. If you registered and did not request higher level access, you will not be able to change some options. So the default is easy. Okay. And some option will be even hidden from you indeed. So second molecule, but for today, you don't need easy access. For today is perfectly fine. So we are also providing the second molecule. So that's the ensemble of structure. Upload it. We don't need to change anything here. And we go to the next menu. If the server will detect problems with your PDB because you, for example, you didn't edit all the atoms of histidine 90 and change it to NEP, it will tell you there is multiple residues with residue number 90. And you will see a reddish error message appearing here on top. So if you get to this stage, it means that the input PDBs were correct. So we do validation on the flight. Now here you can define the information about active residues directly involved in the interaction. So if we go back to the tutorial, we have been visualizing the data. So this is what we just did, okay, step 1 to 4, step 5 next. So we want to define the NMR identified interface as active residue. So I'm going to copy this and paste it here. Okay, you could also select residues from this window on the sequence. I will upload the files in the chat for those of you. Instant successfully. And then also the other one, the ensemble. Okay, so then you have the 2 PDB files if you want to upload. Good. So we have defined those. The neighbors will be automatically defined around those. So the passive residue, so this is automatically defined. This is the default. We can visualize it on the server, and you see now here a view of the structure, and we see again the kind of the donut shape, the binding interface defined by the NMR data. Now we can move. So at the easy level, you might not see those options. Okay, this is a expert level. What what the server has also done for you is to identify the histidine, and in this case, it's only one because the second one was NEP. And we use mole probability to define the protonation state. So it does an analysis of the hydrogen bond network. GROMACS actually does the same. And here it's telling you that histidine 75 should most likely be neutral with the proton attached to the delta position. You can overwrite this, but you need higher than easy access, and you could choose what is the protonation state. So these are two neutral states, but the proton on different location, and this will be the charge histidines. Okay, so you cannot write his plus in the PDB file. This will not be accepted by Haddock. You can also control the flexibility. This is for the refinement stage of Haddock. Here it's automatic, meaning that all the residue at the interface will be automatically made flexible during the refinement, but you will also manually define things or make it fully flexible. That's dangerous because we do a simulated annealing starting at high temperature and cooling down the system, so your protein might partially unfold. Or you might say, I want to have everything rigid, no flexibility at all. But the default is automatic. Some of these might be automatically changed depending on what you're doing. Like if you do peptides, they will usually be defined as fully flexible automatically. Now, second molecule, so I need to define the interface here. The second molecule, this was this list of residue here. Active residues, oh, okay. Ready to go, next. And now we get to the third tab. You see there are three tabs. So in the first one, we input the files, the PDB files. In the second tab, we define basically the active residues. And in the third tab, we can set docking parameters, but we can also upload specific restraints. Like if you have cross-link from mass spectrometry, this is where you would upload those. So some other tutorials are showing you how to do that. So these are some default where we, by default, this is turned on, but we don't need to do that in principle, but we don't need to change it anyway. Also, a good thing to realize is that we are using a united atom force field. Like a methyl group will be represented only by the carbon. So we don't have all the hydrogens described. This is to save computational time. So by default, the non-polar hydrogens, the ones that will not be involved in hydrogen bonds, for example, are removed. If you were to define restraints specific to hydrogens, then you will need to keep those. You see here some of the options that are ab initio options of Haddock. So if you have no data at all, you might want to use center of mass. Do not define the entire protein as active. That's a very bad idea. But use these ab initio options if you have them. You see here this random removal, 50% random removal of restraints. This is something which by default is on. You might not have access to it at the easy level, but you could turn it off if you trust your data. Other type of data, then you see a menu about sampling parameters. So these are the default settings. Depending on what you are doing, you might have to change those. If you do ab initio docking, you want to increase the sampling, but this is simply the default. You see that we don't do this explicit solvent refinement anymore. It doesn't add much to the results, so it's not worth it. Solvated docking mode, so we have a way of dealing with water at the interface, but this only really makes sense if you have good data again. Clustering, so this is how we do analyze the structure at the end. So we use fraction of contact clustering. So we analyze the contact across the interface and we cluster based on contact, not based on RMSD. If you were to do small ligand docking, then you should switch to an RMSD clustering, but the server will do that automatically for you if you define a ligand. While the local version, you will have to do it manually. And then there are many more terms that have to do with different types of restraints that the server can handle. So we can handle hydrogen bonds. They are only intramolecular. We can define dihedral angle restraints to maintain some conformations. These are linked to the symmetry. So if you have a homodimer or homotetramer, you can define C2 or C4 symmetries. So these are the symmetries supported. You can play with restraint energy constants. Some tutorials do request to change some things there. So again, if you want to run those, request higher access. We give higher access to anyone who asks it, but by default, you don't get it. So we might change up to 500 parameters in the interface. So it's, again, if you don't know what something is doing, do not change it. These are NMR type restraints, so dipolar couplings, pseudo-contact shift, relaxation anisotropy, NMR specific restraints, energy interaction parameters. The scoring function is defined here, a simple linear scoring function. So you might change some weight here if you want to, and if you know what you are doing. Some of those are, again, automatically changed depending on the type of molecule. Advanced sampling, so these are used in some scenarios where you can change the simulated annealing protocol. You might change the temperature at which you are running things and the number of steps. If you specify peptides, for example, automatically the number of steps will be increased to allow for more flexibility and more conformational changes. And at the end, you have the analysis where we do, the only analysis is limited to clustering, but you have other options. Full will do more additional analysis, but it costs much more time. So in principle, everything is ready to be submitted here. So you don't need to submit things, but I'm going to do it. But do not click on submit just to not waste computational resources. We have results that are pre-calculated. So is this COVID related? No. And my job has been submitted. One thing I want to show you, so once now it's queued, at some point it will start running, and then you will see the progression. Depending on the load on the server, a run could complete in 15 minutes. It's not very likely these days. It's heavily used. But it also could take several days. It depends very much on the size of the molecule that you are docking, on the number of molecules that you are docking, on the data that you put in. So there are many parameters that affect the computational time. I think I would say that the average running time, if we look at everything, should be probably half a day. An important file that you want to check here is this run parameter file. So we can view it. So this is a JSON file that contains all the parameters for the docking, but it also contains the data and the input data that we put in. So if I scroll down, I see here my atomic coordinates. They are there. I see the list of residues defined here as well. So this is the active list that I defined to the portal, and you see here the surface neighbors that were picked up by the server. So this file is basically a reference of your docking run. So you should always save this file, because this will allow you to resubmit the same run using this submit file interface. Say you want to change to delete only one active residue from the list. What you could do is edit this JSON file, remove the residue that you want to remove, and then submit it through this interface. I'm going to show you how it looks like. So here you give a name to your run, and you upload this JSON file, which is self-contained, and you can submit. So if you do very complex setup where you change a lot of parameters, that's always good to keep. So this will allow you to repeat the docking run at a later stage. You might not get exactly the same results because there's a chaotic nature to the computations that we are doing. And since it's distributed computing, you might be running on different computers. So you will get probably slightly different results, but overall the results should be the same. These kind of files, it's also something that you could provide as supplementary material if you were to publish a paper where you have been using Haddock to generate models for complexes. Okay. Now you see some statistics. There are actually quite a number of, the server is quite busy these days. So there's quite a number of queued requests. Okay, so there's currently 112 runs that are currently being running on a server, and there's a queue of 190 submissions. This is a 2.4 server. I should have higher priority, so at some point my run should start running. We'll see. Okay, so let's go back. So we have submitted things. I explained you the JSON file. We have to keep track of time. So let's go to the results. Okay, so once your run has completed, you should get an email telling you that the run has finished successfully, hopefully, and this will provide a link to the result page. So I'm going to open here the link to the result page. So in the tutorials, there's this link, which is here. It's loading. Yes. So you see status for this job, citations for the paper, acknowledgements. If you had published something important, to put those acknowledgements, because we are getting computational resource for free and we are providing computational resource for free. Can we look at the results image? That's what we are going to do now, actually. Let's see. So now the server tells you, so we clustered 165 structure in 9 clusters. So there are 9 cluster of solutions, which is 82% of the models that have been refined. And what the server is going to show on the result page is only the top 10 clusters. So the job progress you will see here, basically, but there's also a workspace here. If you register, you see a workspace. So let's click on this one just to answer the questions. So I'm going to, so this is my specific workspace, and you can see some of the run that I've been running before. And it's here telling you what's the status. And if you click on that, you go to the result, to the page of that run, and you can see how far it is. But you can also cancel the jobs here. So this will cancel the job. So actually, and you can delete jobs when they are finished. The server is not going to keep all the data. It's going to keep data only for maximum 2 weeks. After that, we delete the runs. So download your runs before that, otherwise you lost it. So the run is not, so you can close the browser, it's not a problem, and come back, you will find back your runs. The only catastrophe that can happen is that there is an total electrical failure on our side, and then the run will be lost. But if things happen on your side, you're safe. So where were we? Result page. Okay. So now for each cluster, you get statistics. So the cluster are presented based on the rank, on the score. Okay. So the top cluster listed on the page will be the best scoring cluster. The number of the cluster has to do with the size of the cluster. It comes from the way we do the clustering. So in this case, the largest cluster is cluster 1 by default, and it's also the best scoring cluster. It's not always like that. Sometimes you get a small cluster which is ranked on top. So cluster size in our hands does not mean that the solution is better. There are other software like PlusPro that use cluster size as a scoring criterion. In our hands, we don't use that. You see a score and a standard deviation, and the standard deviation comes because the scores are calculated on the best 4 model of each cluster. So it tells you the size, it tells you where this cluster is with respect to the overall best ranked model of all the models that you generated. So this one is 1.4 angstrom from the best ranked model, so most likely it's containing the best ranked model. And then you get the component of the score, van der Waals, electrostatic, these are interactions, intermolecular energies, dissolution, restraints, period surface area. And a Z-score, which is only a statistical value. It's just telling you how many standard deviation is this particular cluster from the average of all clusters. So if you only have 2 clusters, one will get a Z-score of -1, the other one will get a Z-score of +1 by definition of the Z-score. Okay, so it doesn't mean you that it's correct or wrong, it's just a statistical measure. You can also visualize online things before downloading to see your complex, turn on the line and zoom in on things and try to figure out, is there a phosphate group? Actually, if we look in this one, you can see here, yes, a phosphate group has been added. Now, if you go down, so you see the second, so the best cluster, 147, the second cluster is 160. So that's quite lower in terms of score. And if you look at the standard deviation, it's not overlapping with the top cluster. So this one is significantly better than the second one. And then you see that the other ones start getting spread. Now, if you scroll at the bottom of this page, you start seeing a graphical representation of the results. So these are interactive plots. So here you will see only the top 10 clusters, but in this case, we only have 9 clusters. They are color-coded based on the cluster. So you can turn everything off and you say, I only want to look at cluster 1. And now you only see cluster 1. The first plot shows you the haddock score versus the fraction of common contacts. That's what we use for clustering. And the second plot shows you the same score, but versus now the RMSD calculated over the interface. And you see here for the fraction of common contact, there is one point here which is at 1. So that's the best model. That's the lowest haddock score, and that's used as a reference. And this same model here is used as a reference to calculate RMSDs. Okay, so it's RMSDs from the best model. You can see that the cluster 1, they are all almost within 1 angstrom of the best model, but you can see that the score is quite noisy. So even if the RMSD are close, the scores are quite noisy. And this comes mainly from electrostatics. So that's something that you have to realize. Small changes in structure might have large changes in energetics. Now you can see the second best cluster was here, and you see it's way higher than the first one, and it's much further away. Okay, you can also, the triangles, by the way, gives you the cluster averages. And these are interactive plots, so you can say, oh, I want to zoom in this region, then you can zoom in that region. And go back. And then you can different, so you have the desolvation energy versus RMSD, van der Waals energy versus RMSD, electrostatic versus RMSD. And here you see distribution for the different clusters. So you see the distribution over the entire cluster populations, where you can compare van der Waals, electrostatic, and restraints. So this is the content of the web page. If you run haddock manually, this kind of analysis will not be done for you. You will have to basically do that manually. There are instructions how to do that. If we go to bonvinlab.org, software, haddock 2.4, manual. Here is the online manual for haddock. And if you go down, you find analysis and scoring. And you see here manual analysis. So it's going to explain you what are the typical analyses that can be done. How would you do violation analysis, post-docking analysis, cluster analysis. So you can look at how to do those things manually. The server has done that for you already. Okay. So we've seen there are 9 clusters. So we've gone basically through this page already, so we don't need to go to that. So now let's look actually at those solutions to see if they make sense. We have about 10 minutes to do that. So if I go back to the server page, the result page, my job is still queued, too busy, not that page. Let's close it. So result page, you can download all the cluster files in one go. So you can download the files one by one, but you can also download them all in one go. So I'm going to download this file. What is it called? So that's, and you can unpack it. And what you see now, so this file contains the top 4 model of all clusters. Okay, so now we can visualize those clusters using PyMol, and that's what we're going to do in the remaining. So in PyMol, there are different ways of loading. So if you have access to the Linux level command, the simplest way is to basically load all of them at the same time. So I'm going to load cluster star_1.pdb. And now you see my PyMol window where I've loaded actually 9 clusters. The best model of all 9 clusters. Okay. I'm going to change the representation a little bit. This is coloring by chain. So you see the 2 chains, but the orientation are still different. So now we want to align all the molecules. So I'm going to select first chain A of cluster 1. Okay, and then you could give this command align cluster 2, 1 to selection, as is done here. And you have to repeat this for the one. But you can, if you know PyMol, you can also go next to the selection here There is an A button. If I click that, there's align, and I say all to this, and then it's done all for you. Let's ditch that window out of the way. Now I've aligned my 9 clusters onto chain A, onto the first molecule. Okay. You can already see that there was some conformal flexibility introduced because the starting point for the green molecule was exactly the same for all molecules, but you see that there are small changes. And you see that here we have quite a number of different orientations. They are all binding in the same region because we define interface residue to guide the docking. So let's look. So this is the best scoring model. And now the second best was, I think was, which one was it? Result page number 3. Okay, so if I compare numbers, so you see, so this, I'm looking now at the top, okay, through the interface, basically. So this is the best scoring model, and this is the second best scoring model. What is the difference between those two, approximately? Any idea? Look where the beta sheets are. You see that? This is the best scoring model. The beta sheet is on this side, and the alpha helix is here. This is the second best. The beta sheet is now on this side, and the alpha helices are here. So it looks a little bit like a 180-degree rotated solution, like a symmetrical solutions. And if you look at the other clusters, this is again rotated 180. This one is slightly shifted, but the same orientation. This one is again a variation of the first cluster. This one again is a rotated solution. This one is a variation of the first one. So you see there are different orientations, but it looks like they are all using the same interface to bind, but they represent different orientation of the different molecules. And this comes because we put data into the modeling process. Now we can look at the data that we put in. So we're going now in PyMod to visualize the active residue that we define to guide the modeling process. So now you see color in red and orange, the residue that we define as active, and they are indeed at the interface. And if you show the side chains for those, you see that they are at the interface. Some of those might seem like they are not making any contact, but remember that they might be over side chains. So if I show all side chains, you start seeing that some of those are contacting. And the important residues are the red ones. So the models, even pretty much the wrong model, seems to satisfy the data, okay? Which is not surprising because we use the data to guide the modeling. So there's one piece of information that we did not use in the modeling process, is the biological function of this complex, which is to transfer a phosphate group from a histidine of one molecule to the histidine of the other molecule. So our model should explain this biological function. So let's visualize those histidines and also this NEP, which is a phosphorylated one. Okay, so I'm going to put it here. So now I'm displaying the histidine as spheres. This is the top rank cluster. And you see, first of all, the phosphate is present. And it's nicely at proximity of the histidine of the second molecule. So based on this model, you could explain the transfer. And if you want to model the transfer, you should probably do, quantum calculations based on the model. So Haddock did generate the phosphate correctly, and the top model puts it correctly. So let's look at the second best cluster, which was quite lower in score. It was cluster 3. And if we look at cluster 3, you see that there is no histidine at proximity of the phosphate. So this is not consistent with the biological knowledge we have on this complex. So this will be another way of validating the results that we have, basically. And you can look at the different other clusters to figure out what they are doing. Now, we could compare with the reference structure. I think we should really kind of finish the tutorial to stick to time. You have a long day, So now I'm going to download the reference structure, which has been solved by NMR. It's here in yellow and orange. I need to do some modification because the numbering between the two was not consistent. Again, you might be lost in the tutorial, but everything is online, so you can take the time to run from the tutorial on your own. And now I'm going to align cluster number 1 onto this structure. I need to visualize cluster 1. Okay. Let's get rid of the spheres. So now you see the docked model superimposed onto the reference structure, which was calculated by NMR. So it's not a crystal structure, it's an NMR structure. Okay. We can calculate, so we superimpose only on chain A, by the way, we didn't superimpose on chain B. And if you look at the RMSD should be reported in this window. So we can calculate what is the RMSD on the, if this worked in the versions, yes. So the RMSD on the second chain on HPR is 2.7 angstrom. Okay, we fit it on chain A and we calculate the RMSD on the second chain. So in Capri, we defined, this is called the ligand RMSD. So in Capri, this will qualify as a, say it's somewhere in between medium and high quality, 2.7. And we only used knowledge of the interface. Of course, in a real situation, you will not have a reference structure, so you cannot calculate this kind of RMSD. So with that, we've come to the end of this tutorial / guided demonstration. Again, it went quite fast. We only had 1 hour to do it. So I hope you have at least get some insight about what Haddock can do for you, what the web portal can do for you. You know, if you have more questions, you can always ask those also using the ask.bioexcel.eu form that we have. All the tutorials are online. All the result page for all tutorials online, so you can visit this again. No, so this is these are classical simulations, so we cannot simulate at a classical level the phosphate transfer. You will have to do quantum calculations if you want to do that. Okay, and that's a different topic, which is probably part of this HPC school. This is why you need HPC resources when you start doing quantum calculations, because they are very heavy. So if there are no more questions, and in the sake of time, I'm looking at Maureen, we should probably start the quiz, right?
Yes, okay.
Yes. So I want to thank you all for attending this lecture and tutorial. I hope you enjoy it. Again, you know where to find a lot of information, so make use of it. You can also, of course, install locally, run on HPC resources. You will have to do more work on your own to do the analysis, but it's also doable. So I enjoy the remaining of the course and good luck with the quiz. I hope you listen carefully to my lecture.
Good, a good closing, Dr. Professor Bongpan. So, okay, now are you ready for the quiz? I hope you're all ready for the quiz. Okay, so quiz time, 10 minutes from now. You can already check your chat box. It's there. Okay, wait a second.